In today's clean label trend, natural sweeteners are often promoted as safer alternatives to sugar. Some are even associated with changes in blood sugar levels in some studies, along with broader metabolic effects that are still being explored. But how much of this is actually supported by human evidence and how much is still based on early research? Before making the switch, it is worth taking a closer look at what the science truly confirms. The intense sweetness of monk fruit comes primarily from a compound called Mogroside V, which exists in only a small proportion of the fresh fruit. Despite this, it has drawn scientific interest for its potential metabolic effects. Animal studies suggest that mogroside metabolites have been associated with changes in fasting blood glucose levels in animal studies. This may involve changes in insulin regulation, potentially through pathways related to GLP-1, a hormone central to blood sugar control and also targeted by certain diabetes and weight management medications, including Ozempic. In mice, high-dose supplementation has been associated with improved insulin response and possible protective effects on pancreatic beta cells. There's also early evidence suggesting these compounds have been studied in laboratory settings in relation to cellular growth processes, including mechanisms involving blood supply. However, these findings are based primarily on animal models, and human clinical evidence remains limited, requiring further validation. Building on these metabolic findings, researchers are also exploring whether monk fruit extract may play a role in more complex conditions, including cancer. In preclinical studies, Mogroside V has been studied in laboratory settings for its potential effects on certain cell growth processes. This effect appears to be linked to the suppression of angiogenesis, a process that tumors rely on to develop their blood supply and sustain growth. In addition to these findings, animal research has highlighted possible anti-inflammatory properties. In models of respiratory disease, mogroside compounds have been associated with changes in inflammatory markers in animal models. For example, data published in the Journal of Food Biochemistry reported improvements in inflammatory markers in asthmatic mice following supplementation. This growing body of evidence may help explain why monk fruit has been historically used in traditional practices for symptoms such as lung congestion and sore throat. While these potential anti-cancer and anti-inflammatory effects are compelling, it is essential to maintain a balanced perspective. The gap between laboratory findings and real-world human outcomes remains a major limitation. At present, most reported benefits, including blood sugar reduction, improved pancreatic beta cell function, and reduced lung inflammation, have been observed primarily in animal models such as mice and rats. Robust human clinical trials that confirm these effects are still lacking. This matters because promising results in animal studies do not always translate reliably to human biology. Many compounds that appear effective in controlled laboratory settings fail to demonstrate the same impact in clinical practice. For this reason, while monk fruit extract shows potential, it should not be viewed as a proven treatment. More rigorous human studies are needed before drawing conclusions about its role in preventing or managing disease. To understand why human trials remain limited, it is important to examine how monk fruit is consumed today compared to its traditional use. For centuries, traditional Chinese medicine has relied on the whole dried fruit to address conditions such as sore throat, lung congestion, and mild respiratory discomfort. It was typically prepared as herbal teas or incorporated into medicinal broths. In contrast, modern commercial products are fundamentally different. Most rely on highly refined extracts that isolate Mogroside V to deliver intense sweetness, estimated to be nearly 300 times sweeter than table sugar. This level of processing changes the composition of the original fruit. Because of this, many of the naturally occurring compounds present in whole monk fruit may not be retained in these products. This shift from whole food to isolated extract could influence how the body responds, potentially altering its overall biological effects. Despite differences in processing, several international health authorities have evaluated this sweetener to determine its safety profile. The U.S. Food and Drug Administration first recognized it as generally regarded as safe in 2010 and reaffirmed this status in 2018. Regulatory agencies in Australia and New Zealand have also reviewed available human data and reported no evidence of adverse effects at intake levels of up to 60 milligrams per kilogram of body weight per day. This range provides a wide safety margin for typical consumption, including use in beverages, dairy products, and tabletop sweeteners. The extract is also permitted in countries such as Japan and Canada, although regulatory frameworks may vary in how they approach high-dose applications. Establishing these intake guidelines helps ensure that increasing demand for alternative sweeteners is balanced with ongoing considerations for consumer safety and public health. Although authorities in several countries have recognized monk fruit as generally safe, the global regulatory landscape is not entirely aligned. In 2019, the European Food Safety Authority published an opinion indicating that the existing toxicity data for this ingredient remains limited. Following their review, the panel concluded that current evidence was not sufficient to fully establish its safety across all intended food uses. This cautious position reflects the need for more comprehensive data, particularly regarding long-term exposure in humans. The assessment also referenced findings from animal studies where certain physiological changes were observed under specific conditions. These observations raise questions about higher intake levels or prolonged use may require further investigation before clear conclusions can be drawn. For this reason, despite its growing popularity, this extract continues to be evaluated closely by some of the world's most stringent food safety authorities. One area of caution highlighted by international food safety panels involves a 90-day study in which male rats were exposed to high doses of Mogroside V. During this period, researchers observed a reduction in both absolute and relative testes weight compared to control groups. Although no immediate reproductive or developmental toxicity was identified, experts noted that the study duration did not cover the full cycle of sperm development. This limitation raises uncertainty about potential long-term effects, particularly under sustained exposure. Because of this, further research over longer timeframes is needed to better understand any possible reproductive risks. Individuals who are actively focused on fertility or who have existing reproductive health concerns may choose to approach high intake with caution. Animal data does not always translate directly to humans, but it remains an important component in evaluating overall safety. Findings from animal studies point to a clear gap in our understanding of how monk fruit extract may affect the human body over extended periods. Experts note that the current toxicity database remains limited, as many studies do not span the full duration of biological processes such as spermatogenesis. Monk fruit has a long history in traditional Chinese medicine, yet the concentrated extracts used in modern products differ substantially from the whole dried fruit found in traditional preparations. This distinction raises important questions about how these products behave in the body over time. High-quality human clinical trials are still needed to determine whether the anti-inflammatory and glucose-related effects observed in rodents can be replicated in humans. Until more comprehensive long-term data becomes available, it remains difficult to confirm potential benefits or fully assess subtle safety risks. Even with ongoing scientific uncertainty, commercial demand for monk fruit continues to expand rapidly across the North American market. The sweetener has positioned itself within mid to premium product categories, largely due to its clean label appeal. Consumers are increasingly seeking zero-calorie alternatives that align with preferences for natural and plant-based ingredients. This trend has created strong momentum for monk fruit as a substitute for artificial sweeteners. Compared with other natural options, monk fruit is often recognized for a more balanced taste profile, which contributes to its growing popularity and competitive positioning against alternatives such as stevia. By mid-2021, an estimated 8,000 consumer products already contain monk fruit extract, spanning categories from dairy and baked goods to beverages and dietary supplements. With continued adoption across the food industry, monk fruit is emerging as a leading candidate among next-generation sweeteners. As the landscape of natural sweeteners continues to evolve, choosing the right option depends on your individual health priorities. For those seeking a plant-based, zero-calorie alternative and who are comfortable with the current level of evidence, this sweetener remains one of several options currently available. Others may prefer a sweetener supported by more extensive human clinical data, particularly when metabolic outcomes are a primary concern. In that context, allulose has gained attention due to research exploring its potential role in glucose-related outcomes in human studies. Each option comes with its own set of advantages and limitations, which makes an informed approach especially important. Monk fruit continues to show promise, yet long-term human safety data is still developing. Staying informed helps you make decisions that truly align with your long-term health goals. Thank you for watching. This content is for educational purposes only. If this changed how you think about nutrition, don't forget to like and subscribe for more science-based insights.